itself. So last week we have already learned about insect hormone. Today we are going to learn about plant hormone itself. So I'm sure you guys know a bit about plant hormones since most of you are taking plant biochemistry. I'm sure there's one or two topic talking about plant hormone itself. And if you look at the Putra Blast in terms of the learning outcomes, what are the things that you should know by learning this plant hormone is at the end of the lesson, you should be able to differentiate the many hormones found in plants. So there are not so many hormones found in plants because it's not as complex as animals or humans. Um, it's just like uh, not that many, and it's just like your insect's hormones, right? But even though there are not so many hormones that we are going to talk about today, but they are important um, in controlling the growth of the plants in terms of the cell turn, in terms of the cell elongation, how basically these plants respond to environmental factors, respond to their surrounding, right? And we are also going to look at the function and regulation of hormones in plant, especially on the auxin, right? The most research plant hormones. And then we are also going to look at how we can use all the knowledge that we have in plant hormones to try and manipulate that to our own advantage in terms of how we can modulate these hormones in agriculture, right? Like processes such as Patinokapi process, producing fruits without seeds. So that is also one of the rules of hormone itself, right? So let me just share my screen. So the lecture for today here.
Recording in progress.
Okay, I have also attached video. If you have time, you can still have a look at this today. But today we are going to focus mostly the first part of it. We are going to looking at auxin hormone. And if you have time, we are also going to look at another hormone which is called gibberellic acid or gibberellin. Okay, I'm going to open up my lecture note for today. So this is going to be the topic of it. So interacting factors in plant development. What basically are the factors that contribute to plant development? So that drive the development of plants. So when we talk about development of plants, it's just like how animal cells are being developed, how insect cells are being developed. It's basically based on cellular proliferation, cellular differentiation, right? Which eventually would bring about cell expansion and cell growth. Okay? And development of plant is regulated by four main factors. Plants can sense and respond to environmental signals. So if you can still remember, I don't know whether you guys learn it in plant biochemistry or not. Uh, the process of photosynthesis is also driven by environmental signals, and we can also see many types of plants. We have C3 plants, we have C4 plants, and CAM plant or CAM plants, which are basically adapting to the environmental signal. So we have plants that grow in an extreme condition. So how do they respond to that extreme condition, and how do they adapt to the extreme condition in order to maintain the livelihood of the plant itself? Such as photoreceptors, which absorb light to send some environmental signals. We can also call this photoreceptor uh, because we know that plants basically have the capability to extract energy from sunlight, which we don't have as humans and also as animals. So plants have the capability to absorb light through organelle named as chloroplast. So chloroplast can also be known as photoreceptors itself, right? Um, and then we have another factor, which what we are going to learn today, hormones. Just like the role of all signal messenger, all right? It delivers, right, in order to impose the effects towards plants. The thing about plants, they are always affected by environmental factors, environmental signals, temperature, the surrounding temperature, the moisture of the temperature, and also the the intensity of the sunlight. So they are always affected by these environmental factors. And last one is enzymes. Just like how we have enzymes in our body, in animals, we also have enzymes in plants, and it's the same function that they have, which basically catalyzes all the biochemical reactions in the development of plants. So basically, enzyme can also work together with hormones in um, driving the development of plants, right? So how do plant develop? Cell division, cell elosion, how it basically drives the cell division and cell differentiation. And sometimes it can also cause, if it is expressed or secreted in high amount, it can cause an abnormal expansion of the cell itself. Okay? So all in all, we can say that there are five major classes of hormones in plants that mediate the plant's growth and development. And these are all the hormones that we are going to look on. So we are going to look at the auxin, cytokinin, gibberellic acid, and abscisic acid, right? And also ethylene. So these are the five major hormones that we are going to learn, we are going to know about when we learn about plant hormone itself. So it's not as many hormones that you might encounter in the plant biochemistry uh, hormone or plant biochemistry subject. So virtually every aspect of plant growth and development is under hormone control to some degree. Plant hormones is also known as phytohormones. It comes from the word phyto and it comes from the word obviously hormones, all right? So the hormones that are produced by plant are called phytohormones. So like any other hormones that we have in animals and insects, these hormones can work solely, right? Work alone, or it can also work together, work collectively, right? Work synergistically in driving one reaction in the plants in our body itself, right? So as you can see, the second statement there, it says that a single hormone can regulate an amazingly diverse array of cellular and developmental processes. It means that the hormones in plant, they don't really stand alone. They basically works together in controlling multiple uh, mechanism or multiple reactions. Or it can either be um, multiple hormones can influence a single process. For examples that we have promotion of fruit ripening by ethylene. Later on, we're going to look at how ethylene is also influenced by auxin, right? And then we're going to look at the regulation of the cell cycle by auxin and cytokinin. Cytokinin is the one that control cellular division. Plant also has cell cycle, just like the one in animals. And then we're going to look at the how seed germination and stem elongation is controlled by um, a hormone named as gibberellic acid. And lastly, um, maintenance of seed dormancy by abscisic acid. And this one here, we are also going to look at it. What is basically means by abscisic acid or abscission. and we are going to look at how gibberellic acid basically inhibit the abscisic acid function and inducing seed germination from a dormant seed itself, right? So this is the and obviously from the look of the fruits itself, it's a tomato plant, right? So if you have looked into um, the many lectures that we have different experimental field or research field, and one of the lectures, Dr. Azrina, one of her research is focusing on how to control the development of a dwarf tomato and a normal tomato plants. So for those, I know that you guys have already got your FYP supervisor's name. You might have some idea what our research are. And Dr. Azrina research is mostly focusing on plants, right? So if, as you can see, we have different parts in the plant itself, and it has different growth rates and different hormones that can affect it. So we start off with shoot tip itself, and we can see that in the soil itself, it has growing roots, right? To basically absorb minerals from the soil itself. And then we can also see vascular tissue, which basically involved in the transportation of nutrients, transportation of hormones in the plant itself. And then we have the tomatoes, right? The fruits, right? How fruits are being developed in plants. And we also have leaves. So leaves are the one that are important in undergoing photosynthesis, in building up the carbohydrates in the plants, and in exchanging carbon dioxide and oxygen between plants and also between humans, right? So it's a very important are basically messengers. It produces at specific locations, and it can be transported from one from the organ that produces it to other organs. And just like the hormones that we learn on in animals, hormones have its own receptor. So it basically binds to the receptor itself, and after that, it can either be entering the cells, or it can just bind to the receptor uh, on the cellular membrane, and then after that, trigger a cascade of reaction that eventually cause changes through the cells itself. And we don't really need a high amount of plant hormones or a high amount of hormones in order for it to induce any changes. It only needs to be secreted at low concentration to basically cause effects towards the targeted cells, right? So it causes altered processes in target cells at other locations. So it basically means that plant hormones also has the capability to be transported from one cell to another in order to impose um, specific reactions to the target cells, right? But one thing about plant hormones is much more simpler, and we can't really, the hormone concept as developed for animals cannot easily be transferred to plants. So we have learned about animal hormones, right? And you guys have learned about multiple types of hormones, and you know that some of the hormones are called peptide hormone. Some of the hormone are called amino acid derived hormone, and we have also learned about steroid hormones. So these hormones here are basically controlled by central nervous system, right? We can see that the steroid hormone is controlled by peptide hormone, and peptide hormone are sometimes controlled by the hormone produced by our pituitary gland, which is also induced by the central nervous system. So that concept is not applied in plant hormones because they don't have um, central nervous system, right? And another thing, like I said before this, plants do not have an efficient transport system as the blood. circulation, like the one in animals, the one in insects. But we look more into animals because we are more developed organism. That's why we have a more complex hormone system, and that's why we have to have an efficient interface transport that hormone from the organ that secreted it to targeted organs. But plants do have a transport system, but it's not as efficient, it's not as developed as the one that we have, which is the blood circulatory system. So we can say that the the hormone mechanism or pathway in plants are much more simpler than in humans. So that's why it is easier to be manipulated for our own advantage in agriculture, right? Despite not having an efficient interface for transport of hormones or transport of nutrients itself, and despite not having central nervous system or CNS to regulate all the hormones simultaneously, plants do have regulated growth. They are able to regulate their own growth because they are living system. They have their own system to regulate growth, and they can determine the steps of differentiation that they need, and they also are capable to have different metabolism in different cells, and they, because of the presence of hormone itself, it has uh, been identified that cells in the plants can also communicate with each other. All right. So the cellular exchange of material in plants is ensured by perforation, or in a layman term, it's called holes of the cell walls at regular intervals. Just like how when you guys learn about photosynthesis, you learn about exchange of the gases, especially on carbon dioxide and oxygen, it occurs through a structure, a whole structure named as stomata or stoma. All right, so this is what it means by this one. The statement here, the cellular exchange of material happens because there are perforation of the cell walls. Just like how in animals we have a membrane cell which becomes our uh, barrier. Okay, permeability barrier, it has its own permeability. It's the same as the cell wall of plants, right? Just like I mentioned just now, these are all the 5 different hormones that we are going to learn about today. So not today, but basically what encompasses in the topic of plant hormone itself. We have auxin, we have cytokinins, gibberellic acid or gibberellin, we have abscisic acid and also ethylene um, hormone. So I have also mentioned to you, think, remember another hormones that I mentioned to you, but these are all 5 hormones that play an important role in controlling the development of plants, the growth of the plants, the cellular differentiation of the plants itself. Right, with the most important one and the most extensively studied being auxin. Right, so we're going to look into the first hormone first, which is auxin, and this is how it looks like. And if you guys can still remember your biological chemistry subject, right? So this is what we call as ball and stick model. We can see that there is a benzene ring here, and then we have a cyclopentene ring here, right? And when we have amine group attached to it, this is what we call as indole ring structure, right? So I believe you guys have learned about your protein metabolism. Um, this structure, this distinct structure here, basically comes from an amino acid named as tryptophan. And we have a substituent here named as acetic acid, and that's why it is called indole 3 acetic acid, or in short, it is abbreviated as IAA. Right? So you can have a look at the description there. Auxin is a plant hormone with multiple roles in plant growth and development, especially in the division of the cells in plants and also cellular elongation when we talk about the shoot tip and also the roots itself. So it is produced both by the tips, the shoot tips, and by the roots itself. And you can see that it is also involved in vascular bundle, also taking place during the production of the nodulation. So what is nodulation? It's basically symbiotic interaction between soil bacteria and plant host, basically for the plant itself to absorb nitrogen as a source of protein, right? So the discovery of auxin started off very early in 1880s by Darwin and Darwin. If you know about theory of evolution, it comes from the same person, Darwin, right? And um, Darwin is so strong about evolution theory. And in Australia, right, this is just a general fact or fun fact. Okay, we have one place um, called Darwin. So because Australia looked up highly to the theory of evolution, right? So that's just fun fact. And Darwin, why is it called Darwin and Darwin? Is basically because the father's name is Darwin, and the other scientist also the other scientist is basically just a son of Darwin itself. So that's why Darwin and Darwin. So they did an experiment, a series of experiments in 1880s, um, where they observed the growth of the coleoptile itself, and they did a different treatment of the coleoptile, right? And they expose the coleoptile through a unidirection sunlight, right? And they observe this one here. In any experiment that you are going to do in the lab, always remember that you have to have control in every experiment. So you define your own control, whether it's going to be a blank control, whether it's going to be a negative control or a positive control. And it's very important, especially when you guys are going to do your FYP um, this coming semester, right? So what Darwin and Darwin did, um, they have a control coleoptile here, and they have a coleoptile with tip remove and a coleoptile that is covered by opaque cap, and another one is covered by transparent cap, and the last one, um, not the shoot tip, but other parts of the coleoptile is covered by opaque shell. And they observe the difference in terms of the growth of the coleoptile itself. As you can see here, the growth of the shoot will usually incline towards the lights, right? And when the tip is removed, they don't see any growth there. And then when the tip covered by opaque cap, it's still growing, but it's not growing towards the light because the light cannot penetrate the opaque cap. And transparent cap, the light can still, or the shoot tip can still sense the light. So nothing happened here. It means that it still is growing towards the light. And this one here, when it puts the opaque shell on other parts of the coleoptile instead of the shoot tip, you can see the growth still happen inclining towards the light itself. And the experiment from Darwin and Darwin, what they concluded that there is a substance that's basically control the growth of the shoot tip itself, but they don't really identify it yet. They haven't identified it yet, but they did uh, drive the experiment further for other scientists called Boyson and Jensen. Right? We're going to look into this later on. But what Darwin and Darwin has found out, it says that auxin, there is a substance that is associated to prototropism, right? It means that the shoot tip is going to grow towards uh, light. inclining towards the direction of the light itself. Auxin was identified as a plant growth hormone because of its ability to stimulate differential growth in response to gravity or light stimuli. So light stimuli, we are referring to the shoot tip, and gravity, we are referring to the root, the growth of the root itself. So the in vitro bioassay in which auxin containing agar blocks stimulated the growth of an oat coleoptile segment leads to the identification of a substance known as auxin, which is indole-3-acetic acid, as the main naturally occurring auxin in plants. Application of indole-3-acetic acid or synthetic auxin to plants caused profound changes in plant growth and development. So much of the knowledge of the physiological role of auxin in plants is derived from studies on how plants respond to excess exogenous auxin. Another experiment on identification of the role of auxins and cellular elongation and so cellular division is this experiment here. All right. So we have a coleoptile, in this case, referring to the statement here, they are using oat coleoptile. All right. So the oat coleoptile, what they did, this is a normal one. They cut off the shoot tip and then they put it on agar block, and after some time the auxin is transported into the agar block, and after that it is placed into the excised tip, all right, excised uh, into onto the coleoptiles that has been excised in terms of the tips itself, and you can see that different growth happens, right? Control without the tip, there's no growth, and control agar block lacking auxin has no effects. There's no growth also because there's no auxin being produced. And this one here, we have an agar block that has been diffused with auxin. We can still see there is a growth, but because there's no shoot tip, we cannot see that it is growing towards the lights. And then we can see here, offset blocks cause curvature, right? We can see that the agar block is placed at the corner, all right, of the coleoptile itself, and we can see that it's following the segments that have more auxin in order for it to grow, and that's why it can cause like a bending kind of thing or curvature itself. So these are basically the experiment that has followed the findings by Darwin and Darwin in 1880, right? So subsequent experiments from Darwin and Darwin, they have found out that auxin play a part in phototropism, an occurrence that involve plants bending or moving away from light itself. So the shoot tip is responsible for directional movement by the plant in response to sunlight, as this is the area where auxin can be found. Later on, I will show you guys what does it mean by this statement here. And sunlight reduces auxin. So auxin basically will be produced in segments of the coleoptile or of the shoot tip that is not exposed to sunlight. It will be produced in high concentration. So the extra auxin present on the shaded sites promotes more cell division and elongation, causing the plants to bend towards the sunlight after this lopsided growth. Right, so let's go to the next slide and see what does it mean by that description in the previous slide here. So this is a coleoptile shoot tip. You can see that there is a production of auxin here, and auxin is more highly produced in segments that are not exposed to the light itself, right? So let's just say that the lights are exposed here, right? So these are the lights. Okay. And this is what we call as shaded area, right? So auxin has the characteristic that it moves away from the light itself. So this is a shaded area, right? So when we have light here, you can see that there are more oxygen here. And when there are more oxygen here, it means that it will induce more cellular division. So which result in more growth, right? So more cells are there, more growth. Right? So that's why this area here is heavier in nature because more cells are there, more growth are happening. So that's why it will then cause bending of the shoot tip towards the light, right? So the bending or the phototropism is because of the presence of auxin itself. And the characteristics of the auxin itself, which basically move away from the sunlight. So this is like a description of what I have already explained to you guys before this, all right? So in 1880s or 1881, Charles Darwin and his son Francis Darwin, what they did, they performed an experiment on coleoptile, right? The sheets enclosing young leaf in germinating grass seedling. So they just grass seedlings, coleoptile, and they with eye observation, right? So the experiment exposed the coleoptile to light from a unidirectional source, one source only, and observed that they bend towards the light. So by covering various part of the coleoptile with a light impermeable opaque cap, the Darwin discovered that the light is detected by the coleoptile tip, but that bending occurs in the hypocotyl. However, the sickling okay. However, the sickling showed no signs of development towards light if the tip was covered with an opaque cap or if the tip was removed. So the Darwin concluded that the tip of the coleoptile was sensing light and proposed that a messenger is transmitted in a downward direction from the tip of the causing it to bend. So this is what Darwin and Darwin did, right? They have a coleoptile from the grass seedlings and they subject it to different treatments. And you can see that this is a control, nothing is being done on the control itself. And you can see that this is the shaded side of the coleoptile where all the auxins occur at high concentration. And this is when the tips are being removed, it's not growing. And tip covered by opaque cap, the statement here. All right. So Charles Darwin and Francis Darwin not just covering the shoot tip, but they also cover different um, different parts of the coleoptile itself. All right. Okay. And this is what they discovered, and they concluded their experiments. All right. They concluded that the tip of the coleoptile was responsible for sensing light. The tip of the coleoptile was responsible in showing a phenomenon called as phototropism, and proposed that the passenger is transmitted in a downward direction from the tip of the coleoptile, causing it to bend. So they know that this bending here is not just because of the light, but it's also because of chemical messenger being produced at the shoot tip and then being transported right from the tip into other parts of the coleoptile itself. So the Darwins followed up this discovery with the experiments. The tips of the coleoptile were covered with a metal foil. This blocked the incoming light and the coleoptile did not grow to its light. This one here. Okay. So Darwin did another experiment and this is what they did. And the growing region of the coleoptile rather than their tips were covered. This one here. Right. The conclusion was made that the growth of coleoptile towards light was controlled by the tip of the coleoptile. And you can see that what I want to reiterate from the experiment itself, Darwin's found out that phototrophism was due to an influence produced in the tip of a coleoptile that moves to the growing region where it causes coleoptile to grow to its light. This influence is what we know today as auxin. So their discovery helped later scientists discover this plant hormone known as um, auxin. So the subsequent experiment was done by Peter Boyce and Jensen, a Danish scientist in 1913, and they did experiment on oat coleoptile, and they showed that the signal was not transfixed on the tip of the coleoptile, but it can right to other parts of the coleoptile itself, which eventually would drive the cellular elongation. Okay? And these are the ones that Boyce and Jensen did. All right. What they did, they uh, they put a gelatin block here, and this one here, the tip is separated by mica plate. Right. Um, they found out that even though they put gelatin block, the growth of the coleoptile of the shoot tip still are growing or still is growing towards the light. So in a way, they have managed to identify that the substance that causes phototropism, the substance that causes the growth of the shoot tip or the coleoptile itself is water soluble. It can diffuse the agar block and be transported into the growing region. So when the tip was separated by impermeable substance, there was no curvature of the stem itself. So this one here, because the substance that they were talking about are soluble in water. When they put mica plate here, it doesn't dissolve in mica plate, so there's no bending of the coleoptile shoot tip. Right. So Peter Boyce and Jensen also have a control. So he cut off the tip of a coleoptile, noticed that it stopped its growth, which is the same observation observed by Darwin and Darwin. So he then separated the tip from the coleoptile with a tiny piece of agar. He observed that the coleoptile grew and curved towards the light. So nothing happened. The coleoptile still is growing towards the light itself, bending towards the light, showing phototropism. So he concluded that the tips of the coleoptile didn't have to be in their normal position to affect growth, and the chemical that control phototropism can move to the agar block. Hence, it means that the chemical is a water-soluble chemical. And then after that, he also did another treatment of the coleoptile itself. He replaced the agar block with butter, right? So instead of putting agar block, it replaces it with butter. So we know that butter are made up of lipids, and he observed that there was no growth, and from this, he concluded that the auxin cannot be transported to other regions of the coleoptile. Hence, this further confirmed the second observation, which the chemicals or the substance they are talking about that affects the growth of the shoot tip is water soluble. So to test if the signal was electrical, he replaced the agar block with pieces of platinum foil, right? The mica plate that, and there was no growth towards the um, light. So therefore, the signal was chemical rather than electrical. So they further confirm what Darwin has observed. But their new finding here, they found out that the chemicals or the messenger that affects the growth of the coleoptile is a substance that is water soluble. All right. And further on, it also leads to other discovery of and other discovery by other scientists, and eventually they have identified that the substance that influence the growth of the shoot tip of a coleoptile is rather a hormone known as indole 3-acetic acid or auxin. So auxins are indole derived molecules. Indole is this one here, structure here. The aromatic structure here, and the cyclopentene structure here with amine group attached to it, right? It can control the division and elongation rate as well as the identity of differentiation state of the cell stuff. So accumulation of and response to auxin have been shown to regulate many growth and developmental processes throughout embryonic and post-embryonic development in plants, right? And it is also involved in seeds and fruit development in plants. So these include tropic growth responses, which is gravity, um, towards gravity or towards light, phototropism. Um, and then we're going to look into another phenomenon known as geotropism, which is towards growing towards the gravity. And these often involve the growing of the roots of the plants. Leaf and flower initiation, root growth, and lateral root formation. So these are effects of auxin. It doesn't just affect the shoot tip growth or elongation, it also affects other things. The roots, the development of the roots, the phototropism, geotropism, and vascular development, and leaf and flower initiation. Right? So how does biosynthesis of auxin occur in plants? So they have identified three different pathways, three important different pathways, and we have another additional pathway also has been identified. The common precursor of auxin in plants is an amino acid known as tryptophan. And if you look at the later slide, you can see that tryptophan itself has a distinct structure here, right? Which is an indole structure. So the common precursor is known as tryptophan or TRP, right? It contains the indole ring itself, and there are several pathways of auxin production. So we have the first pathway, indole 3-acetyldeuxin or IAOX pathway, in which it involves genes like SIR1 and SIR2 that encodes cytochrome CYP83B1. So we have learned about cytochrome P450 um, in what we call as steroid biosynthesis. In plants, they also have cytochrome uh, isoform. M pathway or indole 3-acetamide pathway. And we have the third pathway, the UC and the UC pathway or UCA gene family encoding flavin monooxygenases pathway, which is the main pathway of auxin biosynthesis in um, Arabidopsis thaliana, and it involves the UC gene products of which UC2 protein catalyzes the direct conversion of IPA to IAA, right? So Arabidopsis thaliana. So if you guys have learned about plant biochemistry, this might ring a bell. It's basically a tail crest, mouth, ear crest, or Arabidopsis. It's a small flowering plant native to Eurasia and Africa. It is often used as a reference plant. It was the first plant to have its whole genome sequence and is a popular tool for understanding the molecular biology of many plant traits until today, including flower development and light sensing. So they have found out that Arabidopsis thaliana synthesizes its IAA through a UCA gene family or the UC pathway, where this UC gene produce a protein known as UC2. So this is an enzyme that catalyzes the direct conversion of indole 3-pyruvic acid into IAA or indole 3-acid. And we have another pathway here, which is known as the IPA pathway. So there are four pathways altogether that can produce indole 3-acetic acid with different precursor and enzymes involved. Among four of these pathways here, scientists not knowing just yet which one IAA, because as I told you guys just now, IAA are involved in many um, um, responses of plants. It involved in fruit development, seeding, involved in root development, and also shoot tip development. So it has multiple effects. So it's very hard for scientists to determine which one of the biosynthetic pathway that have been recognized to be the major uh, biosynthetic pathway for synthesis of IAA. And IAA, because the precursor also comes from amino acid, all right? Um, this is tryptophan. So it can also be a precursor of other amino acid, other peptides, and other sugar. So that's why it's a bit complicated to pinpoint that this is the one, uh, this is the major pathway for biosynthesis of IAA. So tryptophan is thought to be the primary precursor for auxin biosynthesis in vivo. So we have um, three pathways are being shown here, but it's definitely four pathways here. All right, we can have the first one, tryptophan being converted into indole-3-acetamide or IAM, and then right away it's converted into IAA. And then the second one, which is tryptophan being converted into indole-3-acetyl-doxim or IAOX, and then being converted to indole-3-acetic acid. And we have another pathway which is IPA here, right? IPA being converted into right away into indole-3-acetic acid. And then we have another pathway here, tryptophan being converted into tryptamine by you know by removing this carboxylic acid group. And then after that, it can go and convert itself into indole-3-acetic acid. So there are four major pathway, or there are four different pathways to produce IAA in plants. Okay. So this one here, I extracted it out from one of the journal articles. It shows you the production of IAA in one cell and the transport of IAA into the target cell itself. Right. So tryptophan as the precursor, it can induce genes such as UG gene and TAA4, TAA1 gene, alright, TAA1 enzyme and UC enzyme here, or UC2 protein, which eventually would convert tryptophan into IAA. And then IAA is being transported out from the cells that produces it through a protein known as PIN protein or PIN integrated protein, right? So this is a protein transporter. It's an active protein transporter. It needs energy in order to transport auxin. You can see here, auxin cannot exit cell freely, but its efflux is mediated by the rate limiting PIN protein, whose polar localization determines the direction of the flux. Later on, we're going to look at the polar localization. What does it mean by polar localization? Polar localization is basically referring to IAA, whether it's in polar form or non-polar form. And then when it is being transported out from the cell that produces it, this is the interface, or what we call as the transport that happens, right? And then it is being taken up by this targeted cell by another influx carrier, again an active transporter known as Lux influx carrier, right? And this IAA will then bind straight to the SCF TIR1. Okay, and read up this one here. Auxin directly bind to the SCF TIR1 ubiquitin ligase and increases its affinity for the auxin IAA protein, targeting these for ubiquitin mediated irritation. So under low auxin condition, AUX IAA proteins inhibit auxin response factors and DNA binding transcription factors, right? So this one here, ARF, is the one that would cause transcription of gene that support cellular respiration. So when the auxin level is low, the RAF is bonded to AUXIAA, right? But when the auxin level is high, it will degrade this AUXIAA and release this ARF, which eventually would cause gene transcription that promotes the cellular division of a plant cell. So that's how synthesis of auxin occurs, transport of auxin occurs, and how auxin would induce the targeted cells. So we're going to look at the polar localization of auxin. What does it mean by polar localization of auxin itself? Right. Okay, let's just have a look at this one here. Like I told you guys, like I mentioned just now, um, auxin is being transported from one cell to another in the plants, and the transport involved in active transport controlled by the polar auxin transport pad system that consists of the protein, pin form auxin transporters. So by regulating expression of the respective gene auxin is involved in its own distribution in various parts of the plant. So we're going to look at the um, diagram or figure. As we know, plant cell also have cell wall and it has a plasma membrane. And just like this, it also have a cytosol or cytoplasm. And you can see that there is a difference in terms of pH between cell wall and cytoplasm. So cell wall is a bit acidic with pH 5, and the cytoplasm is you can call it as neutral, but if we compare it between pH 5 and pH 7, we can say that the cytoplasm is a bit alkaline. All right. So donated auxin can be transported out from the cells into another cells, right? So when it reaches out, the cell wall is acidic, so that's why the auxin will be protonated, and then only protonated auxin can enter the target cell. All right. So protonated auxin enters the cell by active transport, all right, or by diffusion. Okay, and you can see here the difference in terms of the pH is maintained by the hydrogen pump here, right? It keeps on pumping out the hydrogen from the cytoplasm of the cell in order to maintain the pH of the cytoplasm itself, and in order to maintain the acidic pH of the cell wall, so that transport of auxin can occur. This is what we call as polar transport of auxin. It means that when auxin becomes polar or it has charge, it cannot exit the cells. It can exit the cell, right? And it can only be transported in its protonated form. And these are all the active auxin carrier. This is a lengthy description what I have already explained to you guys just now. So we're going to have a look. We're going to read it one first. So according to chemiosmotic hypothesis, what chemiosmotic brain down their electrochemical gradient. An example of this would be generation of adenosine triphosphate by the movement of hydrogen ions across the membrane. Right? So this is chemiosmosis, basically difference in terms of concentration of hydrogen. In this case, when we talk about auxin, it's different between the concentration of hydrogen in the cell wall and in the cell or in the cytoplasm of the cell itself. And it is maintained by hydrogen pumping, that usually is not usually, that is basically an active transport that require ATP for its function. Okay, so according to the chemi-osmotic hypothesis, the difference in pH between the relatively acidic cell wall and cytoplasm maintained by plasma membrane H hydrogen ATPase promotes the accumulation of IAA entering by diffusion inside the cells. So at the more basic pH of the cytoplasm, the majority of IAAH undergoes reprotonation and becomes trapped inside the cell, right? So this one here. So this is the deprotonated auxin, right? It stays inside the cytoplasm, right? So when it is transported in the cell wall, it becomes protonated because cell walls has higher concentration of hydrogen. So when it becomes protonated, then only it can be transported out and transported inside the cytoplasm. So inside the cytoplasm, the pH changes again. So pH 7 and the auxin will be deprotonated. That's what it means by this one here. So this protonated IAA can only leave the cells via active efflux mediated by the specific efflux carrier. So we call it as PIN protein. So the asymmetric distribution of efflux carrier within each cell promote unidirectional polar auxin transport from cell to cell. So transport of the auxin outside of the cell, it requires protein transporter. Inside the cell also it requires protein transporter, or it can also be transported inside with just diffusion. Can read out this one here, the last statement. Auxin efflux is presumably facilitated by multi-component complex consisting of the efflux carrier, we know it as PIN protein, the NPA binding protein, NPV, and third unstable component. This one here, the efflux carrier. It efflux out the auxin, all right? It is an active transport. It consists of a complex of PIN protein auxin influx carrier, the one that carries the influx of auxin inside the cells, facilitate auxin uptake into the cell. Like I told you guys just now, it can either use an active transporter or it can also be just taken up by the cells through diffusion. But this diffusion is also facilitated by influx carrier. So besides influx carrier known as AUX1, other active transport carrier known as ABT binding cassette transporters. So it means that the transporters require ATP in order for it to function, hence it is an active transport. And one of the ABC transporters that is involved in auxin efflux and auxin influx is what we call as PGP or P-glycoprotein family, right? So basically, when auxin is being taken up, can either be taken up by diffusion or by facilitated diffusion, or it also needs the use of a carrier known as auxin influx carrier or AUX1, and then additional influx or additional efflux needed by the plants also is carried out by an ABC or ATP binding cassette transporters known as P-glycoprotein. So that's how complicated transport of auxin is. But one thing that I want you guys to remember is the transport involves deprotonation and protonation of auxin itself. And this is the difference in polarity of the auxin that makes the transport of auxin possible between one cell to another. Right. Okay. Any questions so far? If there is no question, I guess let's take a 5 minutes break. Uh, we'll proceed be 15. That's not really 5 minutes break, but you guys can come back at 3:12, so we can proceed with again oxygen.
Doctor,
Yes.
is there any like simplified way of the pathway, like because I'm a bit confused about the part that whether it diffuse or whether it need require the pin protein or whether it protonated. Uh,
Right. Just give me a minute. Later on, I'm going to explain to you guys. Do it again to you. Yeah.
Recording in progress.
Right. So Nadia Husna just now asked whether there is any simpler diagram to understand the transport of scene itself. Try and simplify it using my OneNote here. I'm not sure whether you guys will understand it, but let's try and understand this. So this is what I put on the slide itself. So we have a figure here, and that this is basically a lengthy description of the oxygen transport itself, right? So before you understand about the oxygen transport, you have to understand the biosynthetic pathway of the oxygen itself and the role of oxygen at the pathway, right? So oxygen is being produced in the shoot tip itself, right? And we established the understanding, all right, that these polypeptide consists of many cells, right? And the one that is producing the oxygen is the one that on the shoot tip itself, all right? And it can be transported from one cell to another, right? So in the cell, in a plant cell, right, it is how plant cells looks like. It has a structure known as a cell wall here, right? Cell wall. And then we have a plasma membrane or plasma cells or cell membrane. It's the same thing, right? All these cell membrane. And we have a cytoplasm here, right? So in plant, there is a difference in terms of pH, difference in pH, okay, between a cell wall here and a cytoplasm. And the difference in pH is the one that makes the oxygen transport possible. Okay, next. So at pH 7, the oxygen is being deprotonated. So in comparison to pH 5 of the cell wall, we can say that the pH in the cytoplasm is a bit uh, alkaline, right? And oxygen in its deprotonated form, it can leave the cells and being transported out by what we call as efflux transporter, right? So efflux here means that effluxing out, removing it out, right? So this is active transporter. So when I said active transporter, it needs ATP, right? So oxygen can only be um, transferred out when it is deprotonated, right? Okay. And then after that, it goes inside the other cells. It can goes like into the cells next to it or neighboring cells. But again, it can either be cellular diffusion or it can be active influx carrier. So this one here is a pin protein, and together with NDP, and another one is an unknown third component, right? So when deprotonated, oxine gets into the cell wall itself, it becomes protonated because it has a lot of hydrogen here because of the low pH. So oxine is being protonated. When it is being protonated, then only it can be taken up by other cells, the neighboring cells, right? So when it is inside the cytoplasm, it becomes deprotonated because of the change in pH itself, right? And proton that is lost by the oxine is then transported out into the cell wall in order to maintain the difference between the cell wall and the cytoplasm. And as you can see here, right, oxine influx carrier, and then we have another ABC transporter. This one here, the transport of oxine inside cells, right? It can happen through facilitated diffusion, right? Active influx carrier, AUX1. So it depends on the condition, right, of the cell, and it depends on the types of the cell, whether the cells is require or whether the cell has the influx carrier, whether the cell doesn't have influx carrier. It also depends on the energy capacity of the cell, right? Whether it has less energy. When it has less energy, it would result to just a facilitated diffusion, right? And if the cell needs extra oxygen being transported, especially for the cells that are growing rapidly, all right, it needs more oxygen inside the cell. It would require the role of other transporter, right? So this other transporter is what we call as ABC transporter or ATP binding cassette transporter. And one of them is known as P-glycoprotein. So this P-glycoprotein will only carry its function when the cells need more oxygen to be transported in. So it helps the more influx of the oxygen in the cells. So that's how transport of oxygen occurs from one cell to another. Does it make sense? Nadira, does it make sense? Nadira Husna?
Um, yes, I tak berapa
You-
faham sangat kata.
Alright, let's just after this you try and read it up again and see whether you can understand it or not. If you still cannot understand it, I will try and I will explain it again to you personally, all right, in the next class. Okay? Because I want to know which part you didn't understand. So we're gonna need like a one-to-one session for that to dissect what you really don't understand about. Okay?
Okay.
So I guess others understand it. Okay, so we're gonna proceed with the rest of the lecture note, I guess. Okay, and here. This is what we have already established, right? Another thing that I think I've already mentioned it to you before this, but I just would like to reiterate to you that the difference in pH, how is it maintained, is through the function of the proton pump itself. This is what we call as chemiosmosis. If you can still remember your carbohydrate metabolism, how ATP is being generated is by difference in hydrogen concentration. So it's the same thing as this one here. The pH gradients or the hydrogen ion gradient is maintained through proton pump. And this proton pump here, it has ATPase function. It has a catalytic function. ATPase means that an enzyme that hydrolyzes ATP. to generate energy or to release the phosphoanhydride bond in the ATP to produce energy. So try to recall back your carbohydrate metabolism lecture note on electron transport chain. The concept is the same, but it's different in terms of in electron transport chain, you have ATP synthase enzyme. And in this polar transport of oxine, you have ATPase enzyme, right? So you have to differentiate what does it mean by ATPase, what does it mean by ATP synthase. But it's a bit easy because one is ACE meaning that hydrolyzing something. Okay. So what is the biological significance of oxine? We have already established that understanding before this, that it basically drives the division of the cells, which eventually cause elongation of the shoot and the roots, but it only is needed at lower concentration. When concentration become higher, the effect reverses and elongation of the root and shoot is inhibited. It's just like a feedback inhibition. All right. Just like how you learn about negative feedback inhibition, positive feedback inhibition, this is the same thing because we know that hormones act through receptor. So when there are too much of the hormone, it would saturate the receptor. Instead of having a positive impact, it would have a negative impact. It's the same thing as this one here. And auxin, it high concentration of auxin would stimulate ethylene production. Ethylene is a gaseous hydrocarbon that is also a plant hormone. So one of its effect is the inhibition of cell elongation. So auxins are involved in the differentiation of vascular bundle. So this is what vascular bundles looks like. We have a dicot and monocot stem. I hope you guys know what dicot means and what monocot mean. If you don't know, you can just refer to your plant hormone notes, or you can just Google it up. What does it mean by dicot and what does it mean by monocot? Dicot means two. So two cotyledons. I've already given you the answer. Monocot means monocotyledon. All right. So auxins are involved in the differentiation of vascular bundles in the plants. They control abscission together with ethylene. It induces beta-1,4-glucanases in pea roots and stimulate the opening of tree buds as well as the rapid growth of a young shoot. And if you look at the notes I have under the slide itself, it says that abscission, what does it mean by abscission? It's a natural detachment of plants, of parts of a plant, typically dead leaf and ripened fruits. And it's basically the plant parts are going through senescence. So it needs to detach the dead leaf. And another note is about plant endo 1,4-beta-glucanases, which have been implicated in breakdown of cell walls during processes observed in normal plant growth and development, including fruits and leaf abscission, grain, germination, and senescence of the plant spot. Right. So in order for abscission to occur, it needs to hydrolyze the cell wall. And that cell wall is being hydrolyzed by an enzyme known as beta-1,4-glucanases. Right. So one of the most common auxin is indole acetic acid. So auxin is basically a group of hormones, but the one that is often identified or occurring mostly is indole acetic acid or IAA, and it can affect the root and shoot tip elongation of the plant itself. So IAA promotes growth in the shoot area of a plant, but at high concentration it inhibits growth, and small amount is just enough to promote growth, whether it's in the shoot area or in the root area itself. So how does auxin drive cellular elongation? Right. So by two effects, right? We have Turgor pressure or Tugger pressure is a force within the cell that pushes the plasma membrane against the cell wall. It is also called hydrostatic pressure and defined as the pressure measured by a fluid measured at a certain point within itself when at equilibrium. Right? Doesn't really make sense if you just uh, read up the statement there. So we're just going to skip to this one here. Right? Okay. So this is a plant cells, and it consists of cell wall. It consists of a cell membrane itself, and we have a vacuole system here where the trigger pressure can be induced. And when auxin is being secreted, it stimulates hydrogen pumping. Okay? Hydrogen pump from the cytoplasm into the cell wall. Remember your polar auxin transport just now. What happened? So if there is an auxin being situated or being placed in the cytoplasm, it would cause protein pump, hydrogen pumping from the cytoplasm into the cell wall, making the cell wall to be more acidic. And when the acidic condition is being achieved, it will then activate an enzyme that is pH dependent that acts optimally, right, at an acidic pH named as expansin. So this expansin will then loosen up the rigid structure of the cell wall, right? And cellulose loosen, cell can elongate because it's no longer in its rigid form. So it is more in a free form now, so it can elongate. And cell elongation is also driven by a Turger pressure, right? So the cells that will undergo elongation or expansion will absorb more water. You can see here, and this would induce what we call as Turger pressure, and then it would also help in elongating cells. So this is what I have already explained to you, and it's written in words. If you don't understand what I have already explained, you can definitely refer to the lecture or refer to the notes. But from the figure itself, you can definitely explain what really happened. So in cells that undergo elongation, that undergo growth and undergo development, we can see that auxin is being secreted. So when auxin is being secreted and being taken up by the cells, the cytoplasm has a pH of pH 7. All right. And auxin would stimulate the um, hydrogen pump so that a pH gradient can happen between the cytoplasm and the cell wall. So the cell wall would become acidic. When the cell wall becomes acidic, it would activate a pH dependent enzyme known as expansin. So why is it named expansin? Because it expands the cell. How it expands the cell is by loosening up the rigid structure of cellulose. All right, cellulose, we know it's a structural carbohydrate and consists of um, glucose as a monosaccharide. Okay, and it basically hydrolyzes the glycosidic bonds between monomers in the glucose in the cellulose itself, and this would result in the loosening of the rigid structure of the cellulose itself, and it can give more opportunity for the cells to elongate. And this cell elongation is also supported by more absorption of water, which eventually would increase the hydrostatic pressure, which would eventually cause expansion of the cell itself. So that's how cellular elongation happens with the help of auxin. So we have to remember auxin is a hormone. Auxin is a signal messenger, right? So it gives signals to other enzymes, right? Like expansin here, and it gives signals to the cells to absorb more water or to uptake more water in order to support cellular elongation. So additional responses of auxin, I've already mentioned to you. Okay, we have excision, loss of leaves, and then flower initiation, sex determination, fruit development. This is how auxin is being manipulated to increase uh, production of fruits without seeds, as Partenocarpy. And then that's it. Okay, so we're going to look at how auxin affects all these processes here, right? So before this, we have talked about auxin and how it affects cellular elongation in the shoot tip and also in the roots. Now we're going to look into how auxin plays another role in these different response of plants. So auxin in leaf abscission. Abscission just now, we know what does it mean. It means that plant sheds one of its part if the fruits are ripening or the fruit starts or the leaf starts to wilt. So it would shed one of the parts. So leaves, flowers, seeds, and fruits are part commonly abscised. So almost any plant part from very small buds and bracts to branches several inches in diameter may be abscised by some species. So it depends on the need of the cell itself, on the needs of the plant itself. So whether it needs to abscise some parts, and it is also one of the defense mechanism in plants, right? So we have um, plants that only abscise or only undergoes abscission annually or biannually, all right? It may show little abscission, especially on the leaf itself. So this is just the one that, just the picture that I want to show you about Jacaranda season in Perth, basically happenings in spring. You see that the flowers is being abscised and is being replaced by a newly formed flowers. This is one of the routes that I usually frequent in Perth when I was there doing my PhD. And it's very, very beautiful. It's like sakura in Japan. So abscission, why does it occur? Why does plant shed one of its parts? Because it can be a process of self-pruning, removing injured, disease, or senescent part. So this is part of the plant defense. And it also permits the dispersal of seeds and any other reproductive structures to support the seed growth in plants. And the third one, it facilitates the recycling of mineral nutrients to the soil. So any dead cells or any dead parts that has been shed by the plants would eventually return back to the soil, and the nutrients can be recycled back, can be absorbed back by the soil. And it's going to be a symbiotic process between plants and between the soil itself, keeping in balance leaf and root and vegetative and reproductive parts, and to maintain homeostasis. Right. So abscission, it has its own zone. This zone is the one that happened. It's where the abscission happens. Separation is brought about by the dislocation of the cell, the wall of a special layer of the cells, named as separation layer. Let me just show you guys here. So we have a plant that consists of the branch and consists of the leaves, right? High oxygen concentration would drive the plant growth, right? And when oxygen level is low, you can see that the plant, the leaves start to wilt, and you can see that there is an increased amount of ethylene production. Okay, so a reduction in oxygen from the leaf increases ethylene sensitivity in the abscission zone. So this one here is what we call as abscission zone, right? The separation layer. And then when ethylene production is high, you can see that it can now shed the wilted leaf. The shedding phase synthesis of enzyme that hydrolyze the cell wall polysaccharide result in cell separation and leaf abscission. So as you can see here, so we have an abscission zone, and this abscission zone we have a separation layer, and how separation layer is being disintegrated in terms of the cell wall is by an enzyme that hydrolyzes the rigid structure of the cell wall. One here. Then only shedding can occur. Right? This is another example that we have. So we have a leaf here, and then petiole is between the branch of the branch and also the leaf itself. All right, we don't really call it a branch, but it's called a petiole. So auxin also plays a role in the abscission of leaf and fruits. Young leaf and fruit produce axin and auxin, and so long as they do so, they remain attached to the stem. When the level of auxin declines, a special layer of the cells known as the abscission layer forms at the base of the petiole itself or fruit stalk. Soon the petiole of fruit stalk breaks free at this point and the leaf of fruit falls to the ground. So the leaf has its own life cycle. So eventually it will turn into a wilted leaf and it needs to be shedded from the petiole itself. And this process is driven by auxin and also another hormone named as ethylene. So at high concentration, young leaf and fruit produce auxin at high concentration, all right, in order to support growth, in order to support cellular division and cellular proliferation. But as time goes by, as the leaf um, grows off, right? And you can see that we have abscission occurs in the abscission layer, right? In this abscission layer, we have a separation layer, and this separation layer is the one that is going to shed from the petiole itself. This one here. So fruit growers often apply auxin sprays to cut down the loss of fruit from premature dropping. So auxin is always used in agriculture to support growth in order to avoid premature fruit dropping or any premature leaf senescence. So this is one experiment to show you the role of auxin in delaying abscission, right? So we have a plant here. Uh, it looks like a pokok semalu kind of thing. I can't remember the English name of it. Pudica, I think so. That's the name. All right. So they did an experiment. This is the petiole here. They cut off the petiole using a blade. They have the one on the left side, the one on the right side of it. The one on the left side, after they cut off the petiole with the blade, they apply auxin. Right. This one here on the right side, they didn't apply anything. And when after a few days later, you can see that the petiole is shedded off from the plant itself. And the one that contain auxin has a delayed abscission process. So it's still intact here. Right? Let me just try and circle it up so that you guys can see. This one here. It's not a very pretty circle there. Right. So it contains auxin. So it delays abscission. Delayed abscission compared to the one that has not been applied with auxin. All right. Okay, so this one here, geotropism, is basically towards gravity. And this happens in the root of plants. Right? So plants exhibit directional growth in response to gravity. So the shoot tips illustrate negative geotropism, but it illustrates positive phototropism, while the root tip exhibit positive geotropism, grows in the same direction as gravity. In roots, the greater concentration on lower side inhibit growth, and the low concentration on the lower side favors the bending of roots downward toward gravity. So we have a roots system here, right? It's following its like growing down, following the gravity. And we have a shoot tip here that shows the phototropism. Both are caused by production of auxin. So this is where the lights are. What it means by that. And auxin also plays a role in maintaining apical dominance. Most plants have lateral, sometimes called axillary buds, located at nodes. Nodes. Buds are embryonic meristem maintained in a dormant state. Auxin maintains it has auxiliary part, auxiliary, auxiliary, auxiliary. It's not auxiliary. It's auxiliary part. So auxiliary is another different meaning, right? So this is what we call as auxiliary bud. Okay. So it becomes or it maintains its dormancy so long as auxin is being produced by the shoot tip here. So when we cut off this one here, the one that produce auxin, no longer auxin, and this auxiliary bud would break into dormancy, would break from dormancy and grow. So most of the plants that don't have um, they cut off the shoot tip here will grow into a bushier kind of plant. See here? If the apex of the shoot is removed, the auxin is no longer produced. This we call the lateral buds to break their dormancy and begin to grow. In effect, the plant becomes bushier. Right? That's what it means by that. So auxin, it basically keep the dormancy of the axillary bud. And when auxin is being, like the apex of the shoot is being removed, it will then produce no longer producing auxin. So just give me a minute. I need to take water. All right. Okay. So I'm gonna share my screen again. Okay. So agricultural use or usage of auxin, seedless fruits, delayed fruit drop, right? So this one here, what we can see is like a delayed abscission, right? So avoiding immature fruit dropping. So at low concentration. And then auxin can also assist in fruit harvesting. It is used for root growth, right? This is what we call as patinocopy. And synthetic auxin, which acts as pesticide, not pesticide. Herbicide, yeah. herbicide. So auxin, beside regulating the growth of the shoot and the growth of the roots, it also regulates root development. Auxin are produced by the pollen tube as it grows through the style and also by the embryo and endosperm in the developing stage. So you guys should know what embryo and endosperms are. This is like a fertilization of the fruits, a fertilization of the plants in producing fruits. So fruit growth depends on these sources of auxin. In some plants, tomato and cucumber, application of auxin to flowers before pollen is mature can promote parthenocarpy, the production of fruits without fertilization, leading to the development of seedless fruit. So fertilization doesn't have to occur. So if you have auxins produced at high concentration, the fruits can be produced without fertilization. Hence, there is production of seedless fruits. We have seedless tomato, seedless cucumber, seedless um, grapes. Right? It's produced because we are able to manipulate the auxin. Right? So high level of auxin doesn't uh, eventually would drive the process of parthenocarpy. And delayed fruit and promoting root growth, we have already looked into this also. And synthetic auxin, which becomes a herbicide, right? Synthetic auxin have been used widely since the 1940s and have revolutionized weed control in agriculture. Their use has decreased production costs by reducing the amount of labor and mechanical weeding needed to grow and efficiently harvest a crop. So we have a synthetic auxin named as 2,4-D or 2,4-D, 2,4-dichloropanoxyacetic acid has a structure related to auxin. It has been used extensively as pesticide or herbicide because it is an expensive and relatively non-toxic to human. It kills the broadleaf dicots, right? It basically kills weeds. This is what happened here. This is the structure of 2,4-D, right? The full name of it is, I can get here, yeah, 2,4-dichloro, this one here, one at carbon number 2 and carbon number 4, 2,4-dichloro, uh, phenol, and we have carboxylic acid group here. And then this one here is an ether group, right? It's known as phenolic acid. So it is the same in terms of the same to auxin in terms of the acetic acid here, right? So auxin is a herbicide. It mimics the genus phase action of the main auxin, indole-3-acetic acid in higher plants. However, they are long-lasting, particularly due to their higher stability. So this structure here, it's stable. A variety of growth and developmental processes when present at low concentration at cellular site of action. So this one here, we have a corn or maize, right? After auxin is being applied at low concentration, it started to grow compared to before exposure of auxin. So at high concentration, um, auxin will kill weeds or herbicides that interfere with the growth of certain plants, right? However, with increasing concentration and auxin activity in tissue, growth is disturbed and the plant is literally damaged. So you can refer to this paper here to read more about the how auxin can act as herbicide and how it can also act as um, growth regulator, right? So it has two functions, and the function is determined by the concentration of the auxin being applied. If it is a low concentration, it would increase the cellular division, cellular proliferation, but at high concentration, it would kill the herbicide. So the farmers would have to play around with the concentration of 2,4-D being applied to the plant cell, right? So this one here, like I said just now, synthetic auxin herbicide finding the log and key to weed resistance. It was published in 2020. It's basically a review article. They sort of collect all the findings and try to find out about how the synthetic auxin can act as herbicide. What are the pathway that they targeted? Basically, they target the same pathway that we have already discussed or we have already talked just now. So the SCF TRR1 gene, and then they have AUXIAA. They basically uh, acts on with, right, anything that interfere with the development of the actual plants itself, right? So we have gibberellic acid. I'm not going to talk about it today because it's been too much, I think. And we're going to continue tomorrow and hoping that we can finish up.
Recording in progress.
So we have learned about plant hormone itself, right? What are plants hormone? They basically are signal messengers, right? Okay, that affect plant growth and development. So we have five classes that we looked into detail. We have auxin, we have gibberellic acid, we have cytokinin, we have ethylene, right? And we have abscisic acid. I don't know whether I spell it correctly or wrongly, right? But you guys can check the lecture notes, right? So plant growth and development are often influenced by environmental factor, right? Environmental factor such as amount of sunlight, right? Amount of sunlight. Okay. Then amount of moisture, right? Right. Amount of moisture and temperature. Amount of moisture, right? Uh, Soil nutrient and temperature. And plant also have receptor. Okay. And one example that we have talked about is photoreceptor, chloroplast pigments. Chloroplast organelle, it has thylakoids that contains chlorophyll pigment. And then we have also looked into the role of hormone itself needed in lower concentration or low concentration, right? In order to cause any effects towards plant growth and development. One of the enzyme that we talk which function together with auxin is what we call as expansin, right? So we have talked about that first, right? And we have also talked about the plant hormones that we have, the five plant hormone that we have, the ethylene, abscisic acid, gibberellic acid, cytokinins, and also auxin. And we know that the plant hormone system is not the same as animal uh, hormone system, right? It's not controlled by CNS, not controlled by CNS, and it doesn't have efficient transportation system or transportation in the space. Transportation system. Just like the one that we have in animals and also in humans, where we have the which equals to blood circulatory system. Um, okay, blood circulatory system. All right. And then we have
Recording
talked so
stopped.
much about the discovery or the history of auxin, right? From Darwin in 1980, 1981. What did they do? And what is the conclusion? What was the conclusion in their experiment? And this is followed by 1913, right, by Peter Poisson Jensen, right, identifying that substance is water soluble, right? So Francis Darwin and also the son is Francis Darwin. The father is Charles Darwin. Okay, and eventually it brings about the identification of the structure of the molecule itself, auxin, which is called indole 3 acetic acid. This is naturally occurring auxin, mostly in plants, right? I've talked a lot on this, which leads to the identification of the role of auxin in promoting growth, right? Especially in showing the phototropism phenomenon. What does it mean by phototropism? What really happened? So you guys have to know that, right? And then I further on explain to you the biosynthetic pathway of the auxin biosynthetic pathway, right? So one is uh, this is not in order of importance or not in order, yeah, not in order of importance. So the first one we have IAO converted into IAA by cytochrome uh, 83B1, right? An enzyme that is encoded by gene protein named SER1 and SER2. And then we have the IAM pathway, or it is called indole 3 acetamide pathway. And then we have another one identified in Arabidopsis thaliana, right? The UCA or UC pathway, right? So we have the UC protein that basically uh, catalyzes the direct conversion of IPA to IAA. That's the first, the port one, and then we have the IPA, and another one that we also have, we call it tryptamine pathway. So this is lack of information on this one here, tryptamine pathway. But more information and on IPA pathway itself, right? And when we have already looked into this, we also look into the transport of auxin from one cell to another. So we have pin integrated protein or efflux of auxin, right? So we have another one, AUX1 as influx carrier, or we can also call it as LEX, right? LEX influx carrier, AUX1 or LEX influx carrier. So the many carrier types that basically influence the uptake of auxin. And auxin bind to what we call as STFEIR1, which produces ARF binding, auxin response factor, which would then increase the production or the protein production that supports growth and division of the cell or proliferation of the cell. Right? So that's what we have learned. And to make things complicated, we also learn about how auxin is being transported from one cell to another. So we call it as polar auxin transport. Why is it polar? Because auxin can either be protonated or deprotonated according to whether it is in the cell wall or cytoplasm of the cells. Right. So the difference in pH that causes the polar auxin transport. So we have cell wall, which is basically acidic, pH 5, and it causes protonation of the auxin. And then we have cytoplasm, which is more acidic than the cell wall itself. So when the auxin leaves the cytoplasm, it is in its deprotonated state. And when it goes into the cell wall, it gets protonated. And this protonated then enters the cells. Right? And then again, it will be deprotonated. And this deprotonation of the auxin will then induce what we call as protein, not protein pump, proton pump, all right? Proton pump. From the cytoplasm, hydrogen ATPase pump, right? From the cytoplasm inside the cell wall, hydrogen. Right? And this different in cell wall also is the one that drive the cellular elongation, right? So at pH 5, there's an enzyme known as expansin, pH dependent enzyme. So expansin, it expands the cell wall, all right? It's an enzyme loosening up the cellulose structure in the cell wall. And with the help of more uptake of water, right, we induce, we increase what we call as a circular pressure, right? We then further expand the cell, and that's how cellular elongation happens. Right. In addition to cellular elongation, cellular growth, division, and proliferation, auxin also has other functions, right? It is also involved in abscission together with ethylene, right? So it is also involved in seed development, flowering process, okay, uh, growth of the roots and the shoot. So this is what auxin does. And we have also looked into what really happened in abscission process, right? You have abscission layer, and in that abscission zone, and in that abscission zone, we also have separation layer, and then we have make it smaller, and we have talked about apical dominance here when auxin is there. But when auxin is not there, there's no longer apical dominance, and the axillary bud here would start to grow, break from dormancy. This one here. So resulting in a bushier kind of plants. Right. And we have looked into auxin in fruit development in terms of Partenocarpy, right? And like I said, it delayed abscission process, it promotes growth, and we have also synthetic auxin, right? Which can act as herbicide. We call it as 2,4-D. And it has a structure that mimic the structure of uh, manipulate gibberellic acid in order for food industry, right? To break down hyaluron layer, in order for brewing industry, alcohol brewing industry. And then we are going to look at cytokinin, how it affects the cell cycle in plants, and going to look into abscisic acid and also ethylene, especially in fruit ripening process. Right? Hopefully we can finish it up tomorrow. Um, all right. Any questions before I dismiss this class? So I think Sopita has already mentioned to you guys, informed it to you guys in terms of the topics that gonna come up with in final exam are up until plant hormones. So plant hormone and the format are basically the same as your test one and also test two. Okay. So any questions? Calling once, calling twice, and calling twice. And before I dismiss the class, I just would like to remind Sofita, if you can remind others who do not come to the class itself, if you don't have 80% of your attendance, you cannot enter the lecture hall. So I told you guys that I'm gonna key in the attendance last week. I mean over the weekends, but I didn't do it. I will try and do it today or tomorrow, and you guys can have a check on it. And if you don't have 80% of the attendance, you have to provide reason to me or to the course coordinator, which is Dr. Izwan, in order for us to allow you to sit for the final exam. If you don't have any valid reason for that, you are not going to be allowed to sit for final exam, and that might cause you to fail the course. Alright? So I hope I made myself clear in telling you about the attendance in order for you to sit for the final exam. Alright. That's it from me for our last lecture.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.